Welcome, everybody, to our monthly Science of Alt Protein seminar. So delighting to see all of you flood in from your different corners of the world. Today, we are joined by Dr. Kyria Boundy-Mills from UC Davis. We're thrilled to learn about her research in yeast, converting inexpensive substrates to oils. It is fantastic to be here with you today. My name is Amy Huang, and I'm the Associate Director of Scientific Ecosystems at the Good Food Institute. To those of you who are new to our ecosystem, welcome. The Good Food Institute is a 501c3 nonprofit that's focused on accelerating our path to a sustainable, secure, and just protein supply via alternative proteins. And that covers the umbrella of plant-based fermentation and cultivated meat, eggs, and dairy. We work to support academics and folks really across the alternative protein continuum. So if you haven't yet already done so, please do consider joining our community at gfi.org/community and checking out our many open access resources, technical publications, etc., for folks who are working to advance alternative proteins at gfi.org. My work at GFI specifically works to support the development of university research and education programs. So if there are those of you in the audience interested in expanding the alternative protein activity at your university, please do feel free to reach out at any time. So delighted to introduce our speaker for today, Dr. Kyria Boundy-Mills, who earned her PhD in biochemistry at the University of Minnesota, studying one gene of one yeast species, after postdoctoral fellowships in microbial ecology at the University of Minnesota and at North Carolina State University. She relocated to UC Davis and partnered with yeast microbiologist and emeritus professor, Dr. Herman Pfaff. She realized how much she didn't know about yeast and absorbed just a fraction of his considerable enthusiasm for yeast, and took over the management of the entire yeast collection when Dr. Pfaff passed away in 2001. As a steward of this 100-year-old collection, she rescued and upgraded the collection, expands it through acquisitions, uses it in her own research program, and amplifies basic and applied use of the collection worldwide. And again, so delighted to be joined today by Dr. Kyria Boundy-Mills. This talk in particular will focus primarily on her research on yeasts that convert inexpensive substrates into oils. This includes substitutes for animal fat and tropical oils for meat substitutes, renewable alternatives to petroleum, and a new class of glycolipid biosurfactants. Her research uses that really rich yeast culture collection at UC Davis that we were talking about before, in addition to 60 years of unpublished data. And we are just absolutely thrilled to have her here with us today to get to learn from her work and her wisdom. So with that, Kyria, I will pass it over to you. Kyria will speak for roughly 40 minutes, and then we'll transition over to Q&A. So if any of you have questions for Kyria, do feel free to drop those into the Q&A section of your Zoom bar. Awesome. Thank you so much for being with us here today.
Yeah, thank you for including me in this seminar series. I'm really excited to share some of our latest results about yeast oils. So today I'll be talking about using yeasts to produce substitutes for animal fats and other useful products. So I will digress and talk about other areas besides animal fat. Just want to give you a heads up. So there are a variety of yeast oils. Most are liquid, as shown here. Some are actually solid or semi-solid at room temperature. So in this talk today, I'll talk a little bit about meat fat, using some material provided by my collaborator, Payam Bahmani, who's a professor in animal science. And then I'll talk about yeast oils in general, the Pfaff collection. I will digress and talk about some of the many other ways that Pfaff collection yeasts are being used. I'm sure there are people on this call who are, in addition to being interested in meat substitutes, are working in other research areas too. So I'm hoping this sparks some new conversations. Then I'll talk about my oleaginous research. I've been in this field for about 12 years. And I'll talk about some of our recent results of yeast fat funded by the Good Food Institute. So first of all, meat substitutes are made out of various plant materials. And then in order to emulate the texture, the flavor, the appearance of the fat, they need some material that's got the right texture, color, flavor, melting point, and stuff. And so what is currently used, for example, in Impossible is coconut oil. And coconut oil is solid at refrigerator and room temperature because of the fatty acid composition. It's high in saturated fatty acids. So that's what's currently used in at least one product. There are also other products that will enter the market soon or are just entering the market. For example, these meatballs, this sausage made using cell-cultured fat and plant-based meats. So there's other approaches in addition that are being explored and developed in addition to using coconut oils. So why do we need fat in these products? Well, fat's necessary for the enjoyment of these products. When we eat fat in meat or in meat substitutes, the fat provides some tenderness. It provides juiciness because of during chewing, the organoleptic, the chewing sensation, and it also provides specific flavors. And there's volatile compounds that are released by the fat, and these triglycerides and phospholipids are responsible for species-specific flavors. That's why, for example, pork, beef, and lamb all have different flavors. So the fat substitutes, we need a variety of them to contribute to these different flavor sensations. So meat fats in various animals have a differing composition of the fatty acids. For example, seafoods such as fish are high in omega-3 fatty acids and polyunsaturated fatty acids relative to other meats. Birds are higher in monounsaturated, and then animals tend to be higher in the saturated fatty acids. So to make substitutes for these various products, a variety of fatty acid composition oils are going to be needed. So this is just another way of showing that. So different oils, such as plant oils, have different composition. For example, canola oil is high in monounsaturated fatty acids. Other liquid oils are high in polyunsaturated fatty acids. Coconut oil and cocoa butter, palm oil are high in saturated fatty acids. And this is what's responsible for them being solid at room temperature, being high in saturated fatty acids. So if you're looking for alternatives, for example, to pork fat or beef fat, just at the category level of fatty acids, palm oil, cocoa butter are additional alternatives, coconut oil. So these tropical oils have similar composition to the beef fat and pork fat. So tropical oils are being used as substitutes in these products, but there are some concerns about these. For example, the palm oil plantations are being built where there used to be tropical forests. So there's environmental concerns over deforestation. There's some market stability issues. For example, a few years ago, Indonesia put a moratorium on new palm oil plantations. There's very few tropical oil production in the US domestically, so we rely on other countries, especially tropical areas. So there's market stability issues. So there's been some exploration of alternative to these tropical oils. using microbes. There's various strategies that have been used to develop these alternatives using genetically modified organisms or non-GMO strategies. And various organisms have been used, ranging from microalgae, bacteria, and fungi, including yeasts. So because I focus on yeast, I'm going to focus on the yeast approach. So just to give a quick background, yeasts are single cell fungi. They belong in kingdom fungi. And they so most fungi are multicellular. There's a couple of branches of fungi that include single-celled organisms. So right now there's about 2,500 species of yeast that have scientific names, but only about 5% of yeast species have been brought into the lab and named. So there's a huge number of yeast species that are just waiting out there in nature, have not even brought into the lab yet, much less named. And just to put this in perspective, about 40 years ago, there was only 500 yeast species that had scientific names. So we're making great progress in bringing yeast to the lab and naming them. But I'm going to circle back to this 1980s figure, so just keep that in the back of your mind. So yeasts are important model organism for basic life science research. I did my PhD studying one gene in one species of yeast. So they're an important organism for basic life science research, and they're also used in a variety of applied areas. Of course, they're used for starter cultures, for food and beverage fermentations, and they're an industrial organism, a chassis organism for production of a broad range of chemicals, including biofuels, food ingredients, medications, all kinds of uses. So they're a very versatile organism. So I'm going to focus on yeast oil. So people have been studying yeast oil for literally 150 years. This is not a new field. Some of the earliest research, in fact, was done in the 1870s. And in this 150 years, about 5% of yeast species, or about 150 species, are oleaginous, which means kind of that the benchmark definition is that it's a yeast that can accumulate at least 20% oil by dry weight. And that's kind of the low end for a high oil yeast. There's some yeast that can be 60, even 70% oil by dry weight. And there's a variety of theories on why these yeasts are doing this. So because these have been known for 150 years, there's been a lot of basic research to understand why these yeasts and how these yeasts are doing this, the genetics, the metabolic pathways, the biochemistry of this ability to accumulate high amounts of lipids has been elucidated to some extent in a few species. And I want to point out this little photo at the top. This is a yeast cell under the microscope that's stained with a fluorescent dye. And these glowing balls inside that yeast cell are these giant droplets of lipids. So this yeast was about 50% oil. So they have this huge amount of oil. Once they start accumulating oil, the cell just keeps eating sugar and converting it. The cells stop dividing and they continue eating sugar. They turn it into oil. So each cell swells up to about 4 or 5 times its previous size. So it's a major shift in the metabolism, and some of the regulation of that has been studied. So like I said, this has been a topic of research for 150 years, but in the last 15 years, 20 years, this has really taken off. So this is a figure from, I didn't make this is a figure from a amazing review article that came out a couple of years ago in 2021. And my suspicion is this grad student or postdoc spent their lockdown months digging into the literature. It has over 800 references, so they really dug deep into the literature. This is probably the most amazing review I've ever seen. And they counted how many publications there were per year going back to the 1970s. And so this across this timeline, in the 1970s and 80s, there were a few dozen per year. Starting in 2005, the number of publications per year has just skyrocketed. So this purple line shows the number of publications per year. So the first point is that there's been an explosion in high oil yeast research. Another thing I want to point out is in this timeline, one little mention here is that in the 1980s, there was some research done on developing a cocoa butter equivalent. And I'll talk about that more on the next slide. And they also, in this review, they also break down where the funding or what type of funding has motivated this research. And a lot of it has been funding to develop biofuels, and a much smaller percent has been used to look at oils for food uses. And so, for example, in my past research, a lot of it was funded for fuels. And so if I had yeast oils that solidified at room temperature, I set those aside as duds because we don't want to put anything that'll solidify into our gas tank. Luckily, with this recent interest in the solid yeast oils, I've been able to pull those samples back because what were duds in my previous research turned out to be hits for my current research. So there's been some exploration of using yeast oils for food applications. For example, during World War I and World War II, when Germany was experiencing shortages of food and petroleum, they had research programs for exploring microbial oils, including yeast, for food uses and for petroleum. And one of the references I saw said that they had tasted some of the oil and it had a pleasant peanutty flavor. So this is using yeast oils for food has been an idea that's been around for quite a while. And again, I mentioned in the 1980s, there was a process using a yeast species, at the time it was called Cryptococcus curvatus, and they developed an industrial technology. They scaled it up to 250 cubic meters. They used a cheap carbon source, which was whey. They achieved really high productivity, 1 gram per liter per hour. That is a high number. But they almost got this process to market, but it ended abruptly because a competing technology using fractionation of palm oil was developed, which so the yeast oil could not compete economically. So this happened in the 1980s. So what I'm going to circle back to this that I mentioned earlier. In the 1980s, again, there were about 500 yeast species that had names. What has happened since then is that 2,000 new yeasts have been named, brought into the lab. There's a huge increase in the number of yeast species, yeast strains that are available for research. So in the last few years, there's been some movement in the market. There's a product on the market from a company called 0 Acre Farms. It is a microbial oil, a liquid cooking oil. There are other companies that are also developing oil-based products for human consumption, solid and liquid products, using a GMO approach, a non-GMO approach. So there is some interest in this area and investment in new companies to explore these areas. So as I pointed out, there was a great review article, and one of the things that this article revealed, the Abel et al. 2021, is that there are some knowledge gaps in oleaginous yeast. So I mentioned there's 150 different species that can accumulate at least 20% oil, but 80% of the publications in this area just use the top 10 yeast species. The other 140 species have only been cited in literature, either 1 or 2 publications. So for the vast majority of species, there's a lot that just hasn't been studied because people focus on their favorite yeast. So what are the phenotypes? What nutrients, what cheap carbon sources can they use? What industrially relevant stresses can they tolerate? What are their overall growth profiles? What's the composition, the fatty acid composition of the oils? the amount and how the amount and the composition changes when you alter the growth conditions. So there's a lot that isn't known, and in order to study the other 140 yeast species, you need access to a large number of yeast species. And luckily, here at UC Davis, we have that. We have the Phaff Yeast Culture Collection. We have over 9,000 strains in our public catalog, and thousands more just in the freezer or not in our public catalog. We have over 1,200 species, so we have about half of the known yeast species. It's the fourth largest public collection of non-GMO yeasts in the world. We have multiple strains of a species, anywhere from 1 to 800 strains isolated at different times in different places from different types of habitats. And again, these are all wild, non-GMO strains. And we also have about 200 species that don't have names yet. And in our database, they're labeled with a Latin prefix AFF. So the source of these yeasts is the ecology research of my predecessor, Herman Pfaff. And also we've acquired yeast collections from other universities. And I've added to the collection from my own research. So about 30% of the yeasts are from some type of food or ag source. About half are from some kind of plants, some are from insects, and then the rest are from various animal environments, seawater, glacial ice cores, all kinds of interesting habitats. We're now in the process of our biggest, it took 100 years to get to 9,000 strains. And we currently have funding from NSF for our biggest acquisition yet. We're adding 5,000 strains in 2 years. So this is a huge addition. We have about 500 species, over about 140 species that were not previously in the collection. So I maintain the collection. I distribute yeast to researchers around the world, and our catalog, our distribution policies, our prices are all on our website. And then I also use the yeast in my own research program. So I can do things with my research program that most labs can't, because I have this amazing resource in my lab. And just a quick side story, I have a visiting professor from Japan in my lab, and he's used 1,000 of these strains. If he had to buy those yeasts at $300 a strain from a public collection, that would be $300,000 worth of yeast. But because he's working in my lab, he can do these projects without busting the supplies budget. All right, so these yeasts are being used by researchers around the world at universities, government agencies, a variety of companies, and they're using them for an amazing range of uses, from basic science, ag and food, health, biotechnology, and other uses. So they're being used in ways that I never would have predicted, but more importantly, the people that isolated and characterized and preserved these yeasts for a specific ecology study, they would have never predicted these future uses. So preserving specimens for future unanticipated uses is the mission of any biological collection. So those are the fields that they're being used in, and then the ways that they're being used also varies. So for example, some folks are using the intact cell, the intact living yeast cell, for example, starter cultures, probiotics, etc. And one quick story, in the 1970s, our collection preserved the yeast that was being used at one of the first craft breweries, New Albion. The New Albion went out of business after a few years. The company they got their yeast from went out of business, but we had that yeast preserved in 1977. So when Boston Brewery wanted to revive this important historical yeast recipe, they were able to get the original yeast strain from our collection. Another way that these yeasts are being used is using parts of the yeast. For example, the primary and secondary metabolites that they produce, structural proteins, enzymes, other antimicrobials. And one example, I have a series of papers about some of our yeasts, in addition to making oil inside the cell, they secrete a new type of biosurfactant outside the cell. And this graph just shows how this yeast is able to reduce the surface tension. It's part of the surfactant activity of this yeast. And then the other way that these yeasts are being used is taking DNA. They're a source of DNA for specific ability to utilize certain feedstocks, produce certain products, or tolerate certain stresses. And I will tell one story about that in a couple slides. So when I'm doing a project, like I said, I have 9,000 yeast strains at my fingertips. I can screen them for some useful property. And so I've done a series of screening projects. You can find out more in my publications. So what we do is we screen this large collection of non-GMO yeasts for yeasts that have some specific property that they're interested in. And we rely on others, on collaborators, or on the companies that want to develop technologies. They do the strain development, the process development. So we just do step one of the project, which is identifying the yeasts that have the most promising base characteristics. So in these screening projects, some of our recurring themes, if we find one yeast that has a property we're looking for, we quite often also look at other relatives of that yeast. Relatives quite often have that property too. If we look at many strains within a species, there's going to be variation within a species. So if we find one strain of a certain species, we can look at additional strains of the same species. We'll often find a superior strain. Also, if the screening technology is really low tech, I can screen hundreds and hundreds of yeasts really cheaply. I will include some random yeasts just from all over the phylogenetic tree, and I'll find some unexpected hits. But if I use a rational selection, which I'll describe in a second, I will have a higher success rate. So the types of data that I use for screening is really varied, and I use all of the sources at my disposal. For example, taking advantage of what type of habitat the yeast came from. For example, a number of years ago, a company was looking for a yeast that could perform a certain function at a low pH. So I included yeasts that were originally isolated from low pH sources, including fruit juice. And our best hit in the whole project, we screened over 1,000 yeasts. The best hit was isolated from lemon juice. So having that original property helped to guide us to hits. We also have a huge amount of data. Before ribosomal sequencing was available, these yeasts were identified by putting them through a long panel of phenotype testing. So we have a huge amount of data. For example, over 1,000 yeasts were screened for ability to tolerate 4, 6, 8, 10, and 12% ethanol. And there were some unexpected tolerant yeasts in that set. And I recently was able to provide these yeasts to a researcher who's studying ethanol tolerance mechanisms. We're also generating our own data. For example, we're using ribosomal sequencing now to identify species. As I mentioned, we have quite a few species that don't have names yet. We were able to provide some strains to a collaborator who was describing and naming a new species, and they allowed us to pick the name. So we named a yeast species after Robert and Margaret Mondavi. So Mondavi aurum is named after major benefactors to UC Davis. And we also can take advantage of outside data. For example, other researchers have sequenced the genome of a lot of our yeasts for various projects. And I will describe quickly one really cool story related to genome sequence. So I'm going to talk about another message, which is that one yeast strain can be used in many different ways by many different people. So this is a yeast strain that I isolated in 2009. The 09 part of the strain ID number means that it was deposited in the collection in 2009. And this was a strain that we isolated from some spoiled olives. We had Maria Marco here at UC Davis and I had a project funded by the olive industry to track down what was causing this spoilage, which was excessive softening. So the olive skin was intact, but the flesh inside had just turned into goo because there was something in there that had excessive pectinase activity. We isolated hundreds of yeast and bacteria, screened them on this plate assay for pectinase activity, and put some of those positive yeasts into a bucket of olives. And this strain, 09448, was able to recreate this spoilage. So we were able to help the olive processors with figuring out what was causing the spoilage and provided some guidance on how to prevent transmission within the olive fermentation facility. In 2011, we sent this yeast and a few others to some researchers in France who were doing some genomics research. They acquired over 1,000 strains from many different collections, including a few from our collection. And this is a figure from their paper published in Nature in 2018, revealing how different Saccharomyces cerevisiae strains are related to and how to each other and how they differ. In 2020, a biotech company in British Columbia was mining this genome sequence data. They found that this olive spoilage yeast and a few others had this property they were looking for, an asparaginase gene. So the reason they were looking for asparaginase is that asparagine that is in fried and baked foods is converted, some of it's converted to acrylamide during the cooking process. If they can reduce the asparagine before cooking, that would reduce this toxic compound. So they took our yeast, they did some adaptive evolution to improve the strain, and last year they produced over 2,000 kilograms of this material and distributed it to food companies to put into their baked and fried foods to see if it could help reduce acrylamide. So this same yeast that was the villain in the olive spoilage story turns out to be the hero in this acrylamide story. So now I'll circle. So that was my digression. I hope that plants some ideas of the many ways these yeasts can be used. So now I'll focus on our oleogenous yeast. I've been working on this since about 2010. And again, I can do this approach looking at many yeasts because we have this amazing collection in my lab. So a lot of the earlier funding was from for biofuels purposes and focused on liquid oils, such as this oil that we squeezed out of some of our high oil yeasts. Some of our contributions in this field is that we discovered 30 species to be high in oil. We revealed the fatty acid composition of about 50 species. We showed that if we change the growth conditions, it alters both the total amount and the fatty acid composition. We profiled what different nutrients, especially carbon sources common in lignocellulosic hydrolysates, so what nutrients they can use, what a few stresses that they can tolerate. And we also took that information to select yeasts and grew some of these yeasts on ag wastes, including corn stover hydrolysate, tomato hydrolysate, grape, many agricultural byproducts. We were able to demonstrate that these high oil yeasts can consume that material and convert it to oil. We also did one little project dabbling with extraction. We partnered with a researcher in the bio and ag department, Dr. Pan, who has a screw press. And so we put powdered yeast in the hopper, this press squeezed it, and what came out was this beautiful red oil. It happens to be a yeast that also makes carotenoid pigments. So some of the information that has been published in various sources, including our publications and 150 years of research, is that different yeast species have different fatty acid composition. So this is taking data from some of our own publications and some other folks' publications. So for example, there's some yeasts that are here labeled A through F that have overall, if you're looking just at the overall categories of fatty acid composition, some of them are fairly similar to these tropical oils or to pork fat or beef fat. There's other yeasts that have, and then these are high in saturated fatty acids. There's other yeasts that are high in monounsaturated fatty acids. So these are some examples. And then there's other yeasts that are high in polyunsaturated fatty acids. So there are different yeasts that are already known to have composition similar to these various types of oils. So that is just looking at the overall fatty acid composition by category. Now, if we look in a little more detail about the individual fatty acids, which are across the bottom here. So in the previous slide, while especially for cocoa butter and coconut oil, those are high in saturated fatty acids. But if you look at the actual composition, so coconut oil, which again is used to emulate fat in some meat substitutes, this is actually high in C12, whereas the actual pork fat or beef fat is high in C16-0. So they're both saturated fatty acids, but they're quite different in chain length. So coconut oil, again, is high in 12-0, and the rest are high in 16-0. I've shown here 5 different strains of the same species on the far right. These are all 5 different strains of Lipomyces tetrasporus. So these are the same species, but isolated in different times from different types of habitats. Even grown side by side and analyzed side by side, they have fairly significant differences in fatty acid composition. And then just looking at the 3 yeasts in the center, these are 3 different species of yeasts in the same genus. So Lipomyces lipifer, Starkii, and Tetrasporus, these have, again, they're different species, they have slightly different patterns. So different species have different patterns, and different strains of the same species have different patterns. And some of these at the fatty acid composition level are fairly similar to pork fat, beef fat. So this is all published data. So we had, based on this promising data, we got some funding from the Good Food Institute to do a 6-month exploratory project. And we divided this into 2 phases. In phase 1, we selected 25 yeasts, and again, we had to use some of our criteria to select which 25. We can't screen all 9,000 yeasts in the collection, unfortunately, but based on the amount of time and labor and cost for this assay, we could do 25 yeasts within this budget. So we selected based on our past work, based on looking at the scientific literature of what yeasts might be promising. We grew these at 50 mil scale and analyzed the fatty acid composition via Payam Vahmani, the animal science department was our collaborator in this project. He did the GC analysis. From phase 1, so we only looked at 1 time point and 1 yeast culture. From phase 1, we picked the 5 most promising yeasts for phase 2, and we grew these at higher volume. We varied some of the growth parameters, and we took multiple time points, and we analyzed the fatty acid methyl ester composition, again by GC. And Just to give a little background, to select the strains, we searched the literature, we looked at our past data, and then we also, it just coincidentally, we had not cleaned out our freezer in a while. We had some of the old oil, little vials of the old oil samples, and based on the fatty acid composition data, we suspected they might be solid at room temperature based on Payam's knowledge of fatty acids. And so we were able to find some of these are some of the old vials that were still in our freezer. We pulled them out, thawed them, and they were indeed solid at room temperature. So, we're supposed to clean out our freezer more often than we do, but luckily, we were a little behind on our cleaning out. And that led us to some really interesting, some candidates. And we also included some species that don't have names yet. So these are new species. The ribosomal sequence doesn't match anything in the public databases. Nobody else has representatives of these yeast species. So those are particularly valuable strains to include. And the results of the fatty acid composition. I consulted with my technology transfer office, and we've decided to move forward with a record of invention on these. So I'm unfortunately unable to reveal what yeasts, what the growth conditions were, what the fatty acid composition is, but I can reveal that we have some promising candidates in the first screen of the first 25 yeasts, and then when we altered the growth conditions, we had also had some promising results. So I can't share with you, unfortunately, today what the exact results were, but I can show you what we'd hoped to find. Again, this is the overall fatty acid composition in real ground beef. If we had a yeast that was fairly close under the initial conditions, and we altered the conditions, we would hope to find something that gets even closer to the composition of real ground beef. And the ideal yeast would be not genetically modified. 99.5% of our yeasts are non-GMO. It would be great if it was a yeast isolated from a food habitat. That would reduce one of the obstacles for commercialization in a human food ingredient. And for licensing reasons, it would be great if this yeast is one of our, about 80% of the yeast in the collection are owned by the University of California. So because they were either isolated by UC researchers or ownership was transferred to the UC when they were acquired. But I can confirm that there were again differences among the different yeast species in both amount and composition of the fatty acids. And that when we changed the culture conditions, we altered the fatty acid composition was altered. All right, so the next steps, we're currently working on filing intellectual property. We're looking at prior art, figuring out where this can fit in. We're writing a manuscript to publish it. We're searching for funding to continue this research. We have a USDA proposal that we submitted a few months ago that's still pending, and we've applied for funding from one UC Davis program, and we're looking to apply from another one. So what we have not looked at yet was some of the properties of these oils that relate to use as a cooking oil. And again, like I said, in my lab, we don't do strain development. We don't do process development. And also, once you have an oil, it has to be put into some kind of structure to emulate adipose tissue in meat. So development of that is something we haven't started yet. All right, so I would like to acknowledge, first of all, Payam Vahmani, assistant professor in animal science, who did all of the oil analysis and provided a lot of background information on what we should be looking for, all the folks who contributed to this work, and our funding sources. And I would be happy to answer any questions.
Fantastic. Thank you so much, Dr. Boundy-Mills, for your fantastic presentation. We've got lots of questions flooding in to our Q&A section of the Zoom bar. So audience members who have not yet gotten the chance to do so, please do feel free to add any questions that you may have accrued over the course of this presentation. Add those questions to the Q&A section of your Zoom bar, and we'll try our best to get around to asking them. Kyria, if you could stop sharing your screen at this point, we'll make it easier for folks to, be able to engage with Q&A. Awesome. There is one question here around whether there are any noteworthy trends about geographic concentrations of yeast in the collection in your lab.
Oh, the geographic, you mean what countries in the world? Yeah,
That
well,
would
we have yeasts from 60 some different countries because they were isolated as part of ecology research. So my predecessors were looking for what function is this yeast in this cactus rot, in this this tree that's infested with a bark beetle. You know, what role is the yeast playing in these specific habitat? So the previous researchers went to the parts of the world where those things were happening. I do want to point out that why that's important, though, for commercial development. So there's a international treaty, the Convention on Biological Diversity, and the Nagoya Protocol, which just in the last 10 years or so has placed some limits on or restrictions on commercializing or any biological material, including microbes that were originally isolated from other countries. That country of origin owns their genetic resources. And so if a yeast was isolated from another country, we have to work within the legal framework of the country of origin if we want to commercialize a technology. So for this reason, when I'm doing these screening projects, if I have multiple strains to choose from, I'll bias my selection towards yeasts that were isolated from the United States, because we're in the US. We have less restrictions because we're in the US. So that's one reason, for commercial reasons, why the geographic origin is important.
Thank you for all of that additional context. There is a couple of questions here about regulatory pathways, and some audience members who are curious to hear whether, for example, a wild non-GMO yeast strain would still need to go through a novel food approval process if used in a food product.
Oh, the approval process. So there's the FDA has a process, the generally recognized as safe, GRAS or GRAS process, to demonstrate. Researchers must demonstrate that this material is safe to use, whether it's a yeast strain, a certain oil, any food ingredient has to go through this process in order to be included in a food product for human consumption. So the GRAS status is for that material for a specific application. And it is possible to do this. Only a handful of yeast species are already described as GRAS, but it is possible to go through the process. And I'll give one example that was before my time, before I started at UC Davis. I've showed some of our pretty red yeasts. There are some yeasts that produce natural pigments that are carotenoids. There's one yeast that makes a carotenoid called astaxanthin, and that is what gives salmon its red color. Farm-raised salmon don't have this in their natural diet like the ocean-raised salmon do, so their diet needs to be supplemented. This Phaff yeast was developed as a technology, as a fish feed ingredient, and it went through the process to be declared GRAS for that specific application. So it is possible at least one Phaff collection yeast has been through that process.
Awesome. That answers a few of the other questions that we had in the pipe as well. There's a question here about the techno-economics of this kind of yeast development. Upon discovery of a potential strain, do you have any insight into how long it typically takes until commercialization and price parity in food-grade yeast oils?
Ooh, yeah. So I know that's something that happens. So I have worked with other researchers on doing that. So I don't do that myself, but working at UC Davis is just wonderful because we have such a broad breadth of expertise. I've collaborated with people all over campus on all kinds of different things. When I get some idea of something I want to explore, like I mentioned the press, putting the yeast in a press to squeeze the oil out, I was able to find someone on campus that had the expertise and the equipment to try that little crazy idea. So there are a lot of people on campus that work on techno-economic analysis. I have written proposals with these people in collaboration. We're actually in the USDA proposal that's currently waiting a funding decision. We're partnering with some other folks at UC Davis on that component. So the proposal it's building on this work that I described, looking at a whole bunch of different yeasts, a whole bunch of different agricultural byproducts, and matching a specific a specific feedstock, finding a partner yeast that can eat that feedstock and make certain products. So matching up the feedstocks with a compatible high oil yeast. And then a big part of that project, if it's funded, will be to look at the economics of the process. So there are people that know how to do that.
Awesome. Thank you so much. Exciting to hear about the related research underway. There is a question here around the growth conditions. You had mentioned that different growth conditions lead to different profiles of fatty acids. And the question here is whether you've noticed different stages of growth also leading to different fatty acid profiles, for example, exponential versus stationary phases.
Oh yeah, that is a big difference. So for most of these high oil yeasts, they are not high oil until late in the growth stage. So when there's plenty of nitrogen and carbon, they have enough material in their growth environment to make the new proteins so they can divide it, so they can grow exponentially. At that stage, these yeasts are only about 10 to 15% oil. So they're a little higher than standard yeast, which are less than 10% oil, but they're like they're like a little bit chubby, but not too much. Once the nitrogen runs out, then the yeast can no longer make protein. They stop dividing. If there's a lot of sugar in their environment still, they'll continue to eat it and they swell up, like I said. And that's when, after starvation of a nutrient, usually nitrogen, that's when they shift their whole metabolism and they start accumulating these oils. And that's when they go from like 10 to 15% oil to these couch potato giant oils, giant yeast cells that are full of oil. So the metabolism undergoes the metabolic pathways undergo this really specific shift that's been revealed for some yeast species.
Very interesting. There is another question here about the crystalline kind of feel and structure of animal fats. So specifically, it seems like animal fats like lard, I don't have direct experience with this, but seems to display a crystalline type feel and structure. Have any of the hard oil analogies that you work with had their microstructures measured? Do they unveil kind of similar crystalline structures, maybe using advanced imaging methods?
Oh, I have not done that myself. I haven't looked at the physical structure of these oils. We've just looked at the fatty acid composition. There are people on campus that are very aware of those kind of issues, and that would be, if our USDA proposal is funded, looking at some of especially the arrangement of the fatty acids on the glycerol backbone in the triglyceride. Which fatty acid is at which of the three positions affects the physical properties. So that is one of the things that we hope to look at if we get more funding in the future.
Wonderful, thank you. This is kind of a related question, more about sensory properties of the yeast lipids. there's just this overarching observation that flavor and mouthfeel in comparison to animal fats is really critical for alternative protein products, and off flavors can be a really big problem with plant oils and meat alternatives. So just curious if you have any insight into the kind of flavor and mouthfeel of the yeast lipids you've been working with.
Oh, so that is something that I have not personally tasted, these yeast oils. Um, the product that's made by Zero Acre, they have not yet revealed whether it's a yeast or a microalgae or some other organism. So I don't know what that is, but there is a product on the market that you can taste if you'd like. Um, and the only other reference I had, I think I mentioned, was that in the World War I, World War II work on yeast oils in Germany, they mentioned it had a pleasant peanutty taste. And so that's about all I can tell you about the organoleptic properties of these oils. I'm looking forward to learning more about that. I'm hoping we'll get funding to do that work in the future.
Awesome, thank you. I suspect, and to kind of reduce the likelihood that I'm asking you similar questions that might just be about research that's in the pipes for future opportunities. Would you say that you have a similar kind of plan in place for measuring the functionality of these oils in like a cooking environment, for example, the pace of degradation or melting, and isn't something that you've looked into to date, but might want to test down the road?
Oh yeah, very much so. Yeah, so that's on my wish list of things I would love to study. And I put a little list on one of my slides: the rheology, the structure, the melting point, smoke point, shelf life, the sensory, the safety issues. So all of those are things that somebody should look at before putting a product on the market. And I'd be very happy to partner with people that know how to do those analyses. And in my lab, we can grow the yeast, we can extract the oil, and then we hand this material over to other people that do the analyses. So I'm very open to partnering with other folks to do some more detailed analysis of the properties of these oils.
Awesome. You heard it, folks. Reach out to Dr. Kyria Boundy-Mills if you are interested in collaborating on these important future research directions. There are some questions in the spirit of collaboration here from our audience as well, specifically around whether you know of any other brands or food developers. You had mentioned Zero Farms, that folks in the audience could research or inquire about more of these oleaginous yeast products in the US or the EU or around the globe. Specifically, they're looking for recommendations on who are kind of credible, established commercial entities developing these processes, in your opinion.
Well, the company that I've been talking with a bit is Zero Acre Farms, and I mentioned they already have a product on the market. Yali Bio, No Palm, Holoferm. There's also, so those are kind of startup, smaller companies, and then a lot of the bigger companies have little research projects in this area. So yeah, so there's a lot of companies that are dipping their toe or in the area, or it's their whole purpose. So there's a broad range of companies that are exploring this area. So it's a really exciting time to have this kind of resource that can contribute to different companies. Like some companies they have their favorite organism that they want to focus on, but they can introduce a genetic material from some of our yeasts into their industrial organism. So that, that's one way that we can contribute. There's other companies that want to have a non-GMO approach. They want to start with a yeast that has the best base properties and then develop that. So we're able to contribute materials in different ways for different approaches.
Wonderful. Thank you so much. In the spirit of that same question, You're clearly such a wealth of knowledge about the research unfolding at UC Davis and the yeast universe at large. And there's a question here about whether there are any specific research groups you know of that might be focusing on the implementation of large-scale production of lipids from yeast.
I'm not sure if they mean academic or
I assumed
companies.
academic.
Yeah, academic. Who's working on large scale production? Um, there's some universities that have these biotech institutes. I got my PhD from the University of Minnesota, and already back a few decades ago, and back in the 90s, 80s and 90s, they already had what they call the Biotech Process Institute, where they could scale up, they could do pilot scale up for companies to demonstrate some technologies. So some of these, there are several universities that have these kind of facilities. There's also a facility run by the Department of Energy in Emeryville, that ABPDU. They have, on a contract basis, they can work with researchers to scale up some of these processes. They have the fermentation and separation technologies. They have very expensive equipment and the expertise to run it. So they can do some of this scale up work in contract with industrial or academic researchers. So there are, companies and there are academic researchers who are utilizing these facilities for scaling up.
Awesome. Thank you so much for that insight. My colleague Renee has also dropped in a database of companies focusing on oil and fat production, which might supplement, those very useful insights. There are a few additional questions here. One is around whether you know of any yeast species that naturally produce omega-3 polyunsaturated fatty acids.
Oh yeah, that was something we were hoping to find. So one of our first papers, it came out in 2013, I believe. We looked at the fatty acid composition of a few dozen different high oil yeast species. And we were hoping to find something that was loaded with omega-3s, and we didn't find any yeasts. There was some that had trace amounts. I'm still looking for that golden yeast that has that property. There are other microbes, other single-cell organisms that do have that property. And I haven't found the yeast yet, but I'm still looking.
I'm hopeful you'll be successful in your search.
Me too.
Seems like a very noble undertaking. In your experience, both maybe in your own lab and other research approaches you've come across, what seems to be the best way to extract the fat from the yeast? What kinds of downstream processing are required? And have you seen any good methods that don't require solvents?
Oh, that's kind of the golden egg that a lot of people are looking for. So that's something I partner with other people to study. In my lab, we can cultivate the yeast, we can harvest the yeast cells, and then we partner with other folks who have the extraction and the separation technologies. So the things that we have personally tried at the academic level, for analysis is what I had needed to develop. In our early work, we compared a few different methods for extracting the oil. Yeasts have this cell wall, so it's got a lot of oil in there, but it's this really tiny little hard little ball. And so getting the oil out is one of the challenges of this technology. So we compared some enzyme, which we looked at autolysis, where if you just soak it in like 50 degrees for a while, the yeast's own enzymes will break down the cell wall. I mentioned putting the yeast through a press. Then what we landed on for the analysis work to extract the oil and to be able to weigh it to see how much oil was there, we ended up just using the standard Folch reagent in a bead beater. So that's what we use at the analytical level to measure how much oil is in there. We did have some fun putting it through the press and seeing the oil coming out of this press. So Dr. Pan's his research focus is on how to get oil out of little tiny seeds like grape seeds, flax seeds. So that's why he has this little bench scale press. So we were able to put powdered yeast into his press that he uses for those other types of oils. And so there are a number of ways that have been tried for getting the oil out of the yeast cells. There's some reviews that have reviewed the scientific literature on how successful these different processes are.
Alrighty. Well, we have just a few more questions here for you, some of which I don't understand fully. So hoping we can decipher this together. There is a question around whether a shift to saturated triacylglycerols, which are solid at room temperature, will affect the growth of your yeast strains.
Ooh, so we grow these at a slightly above room temperature, so like at 30 degrees or so. So hopefully it's not clogging up the little yeast's arteries. We don't want them to have health problems. But so far we've been able to grow these yeasts in our incubators, and they produce these materials naturally. And we haven't seen any slowing down of growth so much when they accumulating these materials. So hopefully it's a natural process that we can take advantage of.
Awesome, thank you. There is also a question here, kind of going back to a question we asked a few minutes ago around the extraction of the fat. This audience member is wondering whether there are any bioproducts that result after that fat is extracted from the yeast, and whether you foresee any applications for those bioproducts or waste streams.
Yeah, so there's that's kind of our model for using these yeasts, is that it improves the economics if there are multiple co-products that can be marketed from one process. For example, we've developed this, I mentioned that we have some of our yeasts that make a lot of intracellular oils also secrete a natural biosurfactant outside the cells. So the multiple products from that process could be the biosurfactant, extracting the intracellular oils. These yeast also are some of these pretty red oils. They make carotenoid pigments. And then also after extracting all the high value materials, the remaining yeast biomass, it's high in proteins. It could have so it could be used for animal feed. It might have vitamins. A lot of these yeasts have sterols in the cell membrane that are important for animal development. Their cell walls could contain valuable glycans. So there's part of our research program is working with a lot of different people to figure out how to valorize all these different fractions.
Makes sense. It's so remarkable to hear about this integrated approach to considering, the entire arc of these yeast species. There's one last question here in our last couple of minutes together. Have you done any research on carbon source specific yeasts? So pentose uptake instead of glucose to be able to utilize lignocellulosic material completely.
Yeah, so that is something that's been a subject of research for quite a few people for many years. So we had one of our papers, I think it came out in 2014, 2016. We pulled out a few dozen different high oil species and profiled what carbon sources they can use. And our list included the prevalent materials that would be in plant material, the breakdown of the structural polysaccharides. So of course, glucose, everybody can eat glucose and fructose, but we also included 5 carbon sugars, pentoses, arabinose, xylose, and then also other materials that could be in there, some organic acids. We included galacturonic acid because that's what you get when you break down certain types of pectin, for example. So that I showed a really quick screenshot of that heat map of which yeasts can eat which of those materials. So that data we've actually published. And we used that data for the paper after that, which was to find yeast that could eat corn stover hydrolysate. So we looked at what was in the hydrolysate. That was mostly glucose and xylose, and a couple others. So we picked yeasts from that spreadsheet, from that heat map, that were best at eating the sugars that are in corn stover hydrolysate. So we have publication on that.
Fantastic. Thank you so much. You are just such a wealth of knowledge. Well, that just about wraps up our audience questions today. I want to give you a big thank you again for being with us to share your information with this global community of scientists. And I would also love to give you an opportunity to share any last thoughts you might have with this community before we part ways. Anything you would like to say to a captive audience of alternative protein scientists and engineers from around the globe.
Yeah. So two things I just wanted to re-emphasize. One is that I'm very eager to collaborate with people around the world. I can only do what I do because I partner with people who do all kinds of complementary types of research. And then I also want to invite people who are in anywhere within driving distance of Davis. Saturday is our big open house, and I'd be happy to chat with you in person about yeast-related stuff.
Fantastic. I hope that event gets tons of fantastic turnout. And I'm sure many folks will be reaching out on the tails of this deeply informative presentation. So thank you again, Dr. Kyria Boundy-Mills, for being with us today. Thank you to all of you for being present with us. And I'm looking forward to seeing you next month for our Science of Alt Protein seminar. Thanks so much, everyone. Bye.
Thanks for the opportunity. Thank you.